And there are also astral microtubules, and those can also be seen on the last page. They are radiating out from the poles and contact the cell cortex, so the outer edge of the cell, and they help anchor and position the spindle in its location for the cell. To discuss the centrosome a little bit more fully, at each spindle pole, it is focused at a centrosome. This is what it's called in most animal cells. It consists of a cloud of paracentriolar matrix, which I call PM, that surrounds a pair of centrioles. And the matrix contains a variety of proteins, as you can assume. The microtubule is dependent on motor proteins that govern spindle assembly. Assembly and function of the mitotic spindle depends on microtubule-dependent motor proteins from two different families, kinesin-related proteins that move toward the plus end, or kind of crawl actually, and dyneins, which move toward the minus end. And there are 4 types of major motor proteins: kinesin 5, kinesin 14, kinesin 4, 10, and dynein. They're important for the spindle assembly and function. You can kind of see a model of this in the next slide, number 13, kind of where they're located in their crawling position. The mitotic spindle needs two poles if it will pull 2 sets of sister chromatids to the opposite ends during anaphase. So there's 2 mechanisms for that. Mechanism 1 depends on the ability of the mitotic chromosomes to nucleate and stabilize microtubules. Excuse me, and it also depends on the ability of the motor proteins to organize the microtubules into bipolar array of minusins at the 2 spindle poles. So it kind of just arranges them in their respective areas. Mechanism 2 is dependent on the ability of the centrosome to help form the spindle poles. The 2 centrosomes facilitate bipolar spindle assembly by providing a pair of prefabricated spindle poles, so it's kind of already there for the centrosome. And the centrosomes are not necessarily a necessity for assembly of the spindle, but it's definitely one of the main mechanisms. Centrosomes duplicate early in the cell cycle, so probably during prophase, maybe a little bit during anaphase. Most animal cells contain a single centrosome that nucleates most of the microtubules, as we just discussed in the last slide, or 2 slides ago. The centrosome duplicates when the cell enters the cell cycle, so at mitosis there are 2 centrosomes so that they can pull the sister chromatids away from one another through the spindles. And it begins at the initiation of S phase when G1 SCDK triggers the cell cycle and initiates centrosome duplication. These centrosomes duplicate and then separate. Then they immediately nucleate the formation of a new centriole so that 2 pairs of centrioles are in the pericentriolar matrix. And finally, the MCDK initiates spindle assembly in prophase. A sudden rise in MCDK activity initiates spindle assembly. 2 centrosomes move apart along the nuclear envelope, and the plus ends of the microtubules between them interdigitate to form their respective position. Centrosome maturation occurs when the amount of gamma tubulin ring complexes in each centrosome increase greatly. So then also increasing the ability of the centrosome to nucleate the new microtubules. And then finally, the multiple motor proteins, as discussed, the dynein and the kinesin, drive the separation of the centrosome in early mitosis. And during prophase, the minus end of the dynein motor proteins at the plus ends of the astromicrotubules provide this major force. Motors anchored at the cell cortex or nuclear envelope, and there's movement toward the minus end, pulling them apart, and MCDK and other mitotic protein kinases are required for centrosome separation and maturation. So now on to the next part of the phase.